Life comes and goes, and even if you can go out and study it again, it's very useful to be able to sort of compare things in your hands among different parts of the diversity of life and also different parts of the world. So in order to do that, we have collections, and we can go to the collections and see, for example, a particular kind of clam or a particular kind of sea star that lives around the world and has different species in different areas, and you can compare them right away. How useful it is depends really on how much material you have in terms of diversity, in terms of spatial and temporal coverage, and in terms of the ways these samples have been prepared. So a good collection will have coverage of much of the diversity of the planet. So in terms of the Florida Museum of Natural History, our invertebrate collections are noteworthy for a number of reasons. One, it's our mollusk collection is the second largest in the country, and our invertebrate collections in general are in the top 5. And it's definitely the most rapidly growing collection in the country because we have a more active field program than anybody else, which means that we get fresh samples that are DNA sequenceable in larger numbers than anybody else does. So we have become a focus of genetic taxonomy because we have the material and we bring in more and more material to allow this kind of work to be done. These are the invertebrate zoology collections of the Florida Museum of Natural History. Both on the right and on the left, you have formed collections that are in pickle, mostly in alcohol, including crabs and sea stars and so forth. And then starting here, you have a lot of dry collections, mostly seashells and land shells and dry material like that. Altogether, the collection holds close to 500,000 lots of specimens, or representing approximately 3 to 4 million specimens. One of the projects that we have recently received funding for from the Sloan Foundation was to barcode our collection. And genetic barcoding is where you take a particular chunk of DNA that's very useful for species identification and use that to identify organisms. And again, you have to build this library so that this sequence of DNA is not just a sequence of DNA, but it's tied to a specimen and an identification. And so we are the major collection in the US for this effort.